Thank you very much for the kind introduction. A personal apology to start with. I am English. I speak virtually no German at all. So like all good English people when sent abroad, we are told from a very early age the best way to communicate with non-English speakers is to shout loudly. If they look at all confused, shout even louder. So I will try that and we'll see how it works out. If you don't. Hopefully, the slides will give you the key messages. The key message, really to get to the end first, because I will talk through some technical details, because that's what I do, is that I'm a toxicologist. I've worked for many years on toxicology problems caused by drugs. I'm particularly interested in rare side effects of drugs. And to be honest, at my time of life, I'm too old to be still describing mechanisms, and I'm far too old to be complaining about people doing the wrong things. At my time of life, I need to be focusing on solutions and way forwards. So that's what I will be trying to focus on in my presentation today. Give you a personal perspective about how we can move away from the issues that have been beautifully set out by the first couple of speakers already today. So I'll be talking about overcoming obstacles to human relevant science with a particular focus on the problems posed by toxicity of chemicals and particularly undesirable side effects caused by drugs. So I will start by giving a personal view of where I believe we are right now, and then where we need to be, and then talk about some of the obstacles that clearly are stopping us getting to where we need to be, because many people have been talking about where we need to be for long periods of time, and the danger is, if we're not very careful, year on year on year, nothing changes. So the interesting part, and the part of the presentation I intend to spend most time talking about, is how we can overcome the obstacles and give some practical illustrations of things that are being done now and some of the major initiatives that are underway that are helping to move things forward. And the bottom line is the solution, I firmly am convinced, is a solution that arises because of communication and collaboration. Nobody has the answer to all of this. No one group, no one organisation, no one country. But when we pull together, all sorts of creative opportunities can arise. So I'm actually very optimistic. I think we're quite close to a major tipping point away from widespread use of animals for many of the procedures they're used for at the moment, simply because we know that the animal procedures really are not giving us the data that we need them to deliver. And we are impatient human beings, and we need science to invest in treating and understanding human problems. So if we're seriously investing as human beings in science that's relevant to humans, then we've got to focus on the human aspect from all of this. So where are we right now? Well, we know, unfortunately, that as we've been hearing in the previous couple of presentations, animal procedures continue to be used routinely to study disease mechanisms, regardless of whether they're the appropriate approaches to use. They're still being used routinely to discover and to develop new drugs, and also to evaluate drug safety. And I have to put my glasses on because I can't see you with my glasses on, but I can see the slides with my glasses on. Apologies. So, particularly in the area of drug safety, animal studies are routinely considered to be the gold standard. So if you ask a toxicologist what's the gold standard to predict and avoid toxicity in vivo, they will say you need to do an in vivo study in animals. So now I'll talk about some examples of where that's complete nonsense. So that's where we are at the present time. The regulatory agencies and science funders have accepted this dogma going forward. I have a lot of sympathy for regulatory agencies, and I know many people who work in regulatory agencies. I have sympathy for them because they have to do something, and the reality is that the animal-based procedures we routinely use to assess safety of drugs and other chemicals have arisen because of catastrophes. And these were approaches that were developed largely through the late 20th century in order to try and make sure that we can avoid problems caused by previous catastrophic drugs that were unintentionally introduced into human usage in spite of the fact they weren't safe enough. So I'll come on to some CNS examples, but a classic example might be in the developmental toxicity arena, thalidomide, widely used for treatment of pregnant women with morning sickness in early stages of pregnancy, in spite of the fact that the drug caused developmental effects in the fetus and had to be pulled afterwards. So we now have an extraordinarily complex set of animal procedures aimed at assessing whether a new investigational drug will cause thalidomide-like developmental effects going forward. And anyway, we know that many animal procedures have limited human relevance. That's where we are right now. We also know that there are many useful human-relevant methods which don't involve animals, which have been described in the literature, and I'll talk about some of those, which have been used and which are being developed going forward. So there's a tremendous amount of good work, and we'll hear about that, particularly for neuroscience, in the later presentations today. So I would argue, although we should be very gloomy about the continued use of the wrong kind of animal procedures or unnecessary animal procedures or flawed animal procedures, actually there's a lot we can do about it. Now, we also know that we have this issue of widespread use of the wrong animal procedures, in spite of the fact that everybody I've ever met who has seriously thought about undertaking experiments believes that the 3 R's principles apply and should only be used. So everybody accepts throughout the whole world that we should only be undertaking animal procedures when they are considered to be necessary because there are no suitable alternatives and because they give us vital information that is particularly relevant to human health. And we've heard good examples in the previous couple of presentations about animal procedures that are being used by people who are seeking to accord with the 3 R's principles, but essentially are contravening them by using useless animal procedures. So that is where we are right now, and we need to do something about it. So because the focus of today's presentation is very much CNS effects, I couldn't resist picking out one example. Those of you who get immersed in regulatory toxicology may be aware that there's a series of mandated studies that are required to be undertaken before any new drug goes into clinical development. And these involve various behavioural measures which are undertaken in animal species. Basically, you look at twitching behaviour, scratching, various other behaviours, and ask whether a new investigational drug causes these effects or doesn't cause these effects. And this is taken very seriously, and the studies are undertaken according to well-defined criteria by anybody who is seeking to take a new drug into clinical development. Now, a paper came out a couple of years ago from some very eminent individuals, some of whom were my ex-colleagues in the pharmaceutical industry, which assessed whether the outcomes from these rodent neurofunctional assessments had any relevance at all to subsequent behavioural effects that could be caused by the investigational compounds when the investigational compounds were actually progressed into clinical trials. And the answer is quite shocking. There is no concordance at all between the outcomes from the rodent-based behavioural neurofunctional studies that are undertaken in animals and the undesired pharmacological effects that they're intended to predict in humans. Now, this is 2 years ago. The way that the paper is described doesn't set it out as bland, as aggressively as I have, but I invite you, there's the reference, read it for yourself, and if you want to disagree with me, disagree with me. So I would argue this is a really good example of how we are doing things at the moment. We really shouldn't be doing them. It's a complete waste of time. We know that neurotoxicity is not what people think of as the most important toxicity that people need to be concerned about before we take a new drug into clinical development. The more frequent toxicities that often cause clinical complications would be cardiovascular toxicity and liver toxicity, and I'm particularly interested in liver toxicity. But if we look at neurotoxicity caused by drugs, it can be a major problem. Now, I don't know how many people in the audience today have been involved in drug development and worked in pharmaceutical industry, but the reality is that many of the drugs that get through to licensing and are considered to be effective, many of those drugs have to be discontinued subsequently. And there's a really powerful database that's been described a couple of years ago, again, a reference here, which lists over 500 drugs, licensed drugs, that were withdrawn or discontinued from clinical usage. So this is a large number of drugs. And half of those withdrawals were due to toxicity, and this circle indicates causes of toxicity, and you can see that neurological effects, severe neurotoxicity, is nearly 10%, 9% of the problems. So this is a big issue moving forward. It's really difficult to understand. I work on liver toxicity, which is difficult, but neurotoxicity is much more difficult, and we need to find a way to deal with this. These are drugs that were withdrawn after going through clinical trials, after being considered appropriate to be licensed, and after being used in human beings. So these are drugs that have passed through all the conventional toxicity tests in animals and other procedures. So there's a big gap. We need to do something about neurotoxicity moving forward. And anybody in industry would agree with this. It's not given a top priority by people in industry at the moment because it's difficult and we are struggling. And when I say we, I would include myself in that because although I'm not in industry anymore, I continue to consult with industry and I continue to try and help to solve the problem moving forward. So we know there are lots of neural cell models, and I'm not going to try and talk through this, but there are plenty of neural cell models, and there's a lot of interest in using neural cell models to try and generate data which is predictive of neurotoxic effects caused by drugs in humans. And I know that there's interesting progress that's starting to be made. I don't believe we're there yet, but I know this is a topic, neural cell models, that will be talked about later, and the toxicological application is definitely something where I think we will see some substantial progress occurring in the relatively near future. So, anyway, that's where we are. That was the doom and gloom bit of the presentation. Where do we need to be? I believe we need to be, this is my vision of where we need to get to, we need to be in a situation where human-relevant non-animal methods can enable accurate and effective investigation of human toxicities and particularly human diseases. If we have good human-relevant models, then we'll be able to identify and develop good, safe drugs. And we also need to have human-relevant non-animal methods that can be used reliably to take decisions within industries and by regulatory agencies on whether to bring a new drug forward. And that's the vision of where we need to get to. We need regulatory guidelines that mandate use of appropriate non-animal models, and we need scientific researchers and funding agencies to be focused first and foremost on human-relevant models. I don't want young scientists to be thinking, I need to work on a transgenic primate model of, say, Alzheimer's disease, for the sake of argument, following the previous presentations. I want a new researcher to be thinking, I want to work on Alzheimer's disease, gain new insight, and I want to use the most appropriate model, and this is the most appropriate model. It uses human cells cultured under physiologically relevant organotypic conditions, and the data from this model I know how to understand to gain insight into human disease and human toxicities. That's where we need to be. That's where the science needs to get us to going forward. Now, for neurotoxicity and neurosciences, we are not there yet, and I don't think we're very close to there yet. In other areas of toxicity, particularly, I think we're getting really quite close to the tipping point where we are going to move away from reliance on animal-based procedures and move into reliance on human-relevant models. For cardiovascular toxicity, there's a big international initiative which is developing something called the SIPA panel, which is really gaining a tremendous amount of traction within industry and regulatory agencies. It's been widely recognized and appreciated that cardiovascular toxicity caused by drugs in humans is very poorly predicted by animal studies, but there are a whole variety of mechanistically relevant, human-relevant methods that have been developed and refined. So for cardiovascular toxicity, that's coming. For liver toxicity, my favorite area, I'll come on to some examples. So what are the obstacles that are getting in the way of things? Well, we've heard already that a lot of our current understanding of in vivo biology and physiology is based on in vivo investigations that came largely in the last century from studies undertaken in animals. And it also, a lot of our understanding of in vivo biology in humans is based on what we can investigate in humans. So obstacles hindering change, we've heard it's difficult to do studies in humans. We can do some things in humans, but we can't do everything in humans. So in principle, working with something other than adult humans, particularly with advanced diseases, would give us big advantages. We know that human diseases and toxicities are complex, And there's an enormous amount we don't know, and no doubt that's why we've got terrible models of, say, Alzheimer's disease at the moment, or various neurodegenerative diseases. Apologies if I've offended somebody in the audience who's going to speak later and say we've got a really good model that I'm not aware of, but my feeling, having read the literature as a non-expert, is that the models that are conventionally used to try and understand human neurodegenerative diseases and human neurotoxicities really are a long way from where we need to be. We've got lots of in vitro models. In vitro, for those of you who aren't experimenters like me, this means that we're working with cells or broken cell preparations. In vitro comes from in glass. So nowadays we do things in plastic, but in fancy devices. But it's non-animal. So there's lots of in vitro models. But we've really got to struggle still in integrating together data from various in vitro models. And as soon as you break down a complex organism into cells and broken cell preparations, then you lose a lot of the relevant physiology and biology. So how do you put it all back together again? So that's tough. It's difficult. People are struggling really hard on it. And I've spent many years wrestling with this. But this is no doubt a major obstacle that we need to surmount. We know that the scientific community is conservative. And all of us who are scientists know what we know. And we're all inherently somewhat nervous about talking and working in areas that we don't know very well. So some of the really powerful innovations and developments in vitro models and complex mathematical approaches to integrate model data from in vitro models, well, these are people who don't run animal experiments. The kind of people who run animal experiments know how to run animal experiments. So, you know, those are conservative people. Regulatory agencies we know need to be risk averse. They will not stop undertaking animal experiments until there's overwhelming evidence that's accepted by the scientific community and mandated by regulatory guidelines that we can generate equivalent or better quality data in other ways. So the regulators aren't going to be pushing this. The regulators quite rightly need to be at the back of the queue. And I think that's correct. If I'm taking a new drug, I want conservative regulators to assure me that it's as safe as it possibly can be. So we've got to take account of that. I'll also, this final one is a personal complaint, really. I've worked in this area for a number of decades now, and I've got infuriated many times talking and arguing with very bright colleagues who are very, very quick to criticise and very slow to innovate. So I've got tremendous respect for anybody who's looking to innovate and propose a new philosophy, a new model, a new way of thinking, because I've got the bruises to show for it. You know, as soon as you propose something new, people come and try and beat you up because they say, we tried that before, it failed, so on and so forth. Anyway, these are obstacles, but there are ways forward. So just a couple of words on models. This is really very important. If you talk to biologists, every biologist we'll go back one. Every biologist inherently loves these really sophisticated organotypic models. So these are some cells cultured in a device where you can put fluids going through. And this is one of the early prototypic liver-type devices. And these are wonderfully complex devices, and in principle they reproduce an enormous number of the interactions that could occur in vivo in tissues and in cells. So fantastic biology, incredibly difficult to work with, really difficult to get robust data, and very difficult to get quantitative data on large numbers of compounds in a way that you need to assess potency. So these are difficult devices, everybody likes working with them. Then we've got very simple devices with simple cell systems, and the more complex the system, the greater the cost. The more complex the system, the less data you can get back from it. And if you talk to toxicologists in particular, if you get 10 toxicologists around a table and you ask for their views on which in vitro models we should be working with, you get at least 50 different responses. So it's all over the place. You know, and we need to acknowledge the fact that when we're moving away from animal-based experiments, and we're talking about human-relevant in vitro-based approaches, it's really difficult when there aren't consensuses going forward. And when we're thinking about which non-animal human-relevant model we should be working with, the first question we've always got to address is, will the data from this human-relevant model actually be mechanistically relevant or not? And we need to have an answer to that, and we need to have a way to argue that these new data are mechanistically relevant, and we can interpret them, we can make sense of them. I would say, equally as important as that, can the data from the in vitro model be generated robustly, reproducibly at the throughput that's necessary? with the turnaround time and cost that's necessary to actually be used in practice. If you're working in the pharmaceutical industry, there may be enormous amounts of resource, but when you're trying to test a new molecule, there's actually limited amount of time before the chemists have made another molecule. So these bits about pragmatism are really very important. And robustness is vitally important if we're talking about using data for regulatory purposes, because if we can't get the same numbers routinely every time we run the assay, or we can't get the same numbers in the same ballpark when we run the assay in different labs in different parts of the world, then how can we take decisions on the back of it? So it's difficult. We've already heard about complex endpoints. There are an enormous number of endpoints that can be measured, and biologists are complicated, and now they can do gene expression profiling, so they can measure hundreds and thousands of things, and we can use high content biology, and we can measure so many endpoints that your head gets sore thinking about it. And then we really struggle to interpret data from even single assays. And then when we think about mechanistic relevance, mechanisms in vivo usually involve multiple biological processes. All toxicities, all complex diseases are complicated. So we always need multiple in vitro assays, and we've got to have some way to be able to integrate the data and to make sense of them. So these are real challenges, and this is why the field of moving from animal experiments to human-relevant non-animal experiments is moving so apparently slowly. So what can we do to make progress? Well, be pragmatic about it and not be overly ambitious. Bite-sized goals are vitally important. My argument about 15 years ago, when I seriously got involved in this, working for a pharmaceutical company, AstraZeneca, was that we needed to have tangible targets, and we needed to start selecting some models that we felt would be human relevant, but would also give us data in a reasonable time frame. And then we needed to pick what looked like the best models using pragmatic selection criteria and generate some data ourselves, because the data we needed to take decisions simply wasn't available from the literature. There were lots of models, but there weren't the data that we needed. So these are still obstacles. Anybody coming into this, no two people will agree on the best way to tackle all of this, in spite of the big initiatives. This is a cartoon which I put together a number of years ago for liver toxicity, but it applies to any adverse effect caused by any chemical or any toxicity caused by any chemical. The first thing is, well, you could call it a drug, you could call it a molecule. You'd only get an adverse effect if you expose a biological tissue to the drug or the chemical. And there's always metabolism, what we call ADME, absorption, distribution, metabolism, excretion. That always happens. And then, in order to get toxicity, there always has to be some sort of chemical stress within target cells. I'm interested in the liver, so I was particularly interested in chemical stress within target cells. And after that, chemical stress caused by drugs or metabolites, it always gets horribly complicated, and there's a load of stuff going on which you don't understand very well. And that's where we are at the moment. But actually, there aren't that many ways that drugs and other chemicals cause chemical stress to target cells. And we understand an enormous amount about disposition and ADME-based properties. So our simplification about 15 years ago was to say, well, if we start at the top end and we worry about this stuff, can we actually help to start to identify bad drugs which have the potential to cause toxicity while ignoring the complexity in the middle, the human-relevant-based approaches? Now, this is heresy for toxicologists. Toxicologists don't like to make assumptions, and they don't like to take out the bit in the middle. But actually, this is the philosophy that's been used for predictive toxicology purposes. Essentially, we're worrying about the drug or the chemical. We're selecting pragmatic endpoints from some pragmatic, mechanistically relevant stressors that chemicals can cause to cells. And we're saying, if we do that, can we make the first step of a hazard assessment, and then can we bring in some sort of data interpretation to get us to risk? So, believe it or not, it does appear to work, and that's the basis of the SIPA panel for cardiovascular toxicity, and that's the basis of the hazard identification approach that translated into a risk assessment approach, thank you, for liver toxicity. So these are some cell systems, and these are some chemical insults. If anybody likes technical details, which I do, I've got more than enough technical details, but I haven't got time to go into it today, and I know most of the audience are not technically inclined. But please get in touch, and I'll talk you to death about technical details. These are just some of the articles, and this is a personal view on it. But the bottom line is, this was a very simple approach, but actually it worked in practice. What we needed to do was have multiple assays. We had to have some cunning way to link together data from multiple assays. And one of the endpoints we were concerned about was formation of chemically reactive metabolites, which are really important for toxicology, but it's only part of the problem. and we needed to take account of reactor metabolite formation in a way that could be quantified and took account of daily exposure to drugs. So that was one axis here, and the other axis was adding together multiple in vitro assays. But the good thing is that we tested 36 drugs, and 9 non-toxic drugs that were widely used with very good safety profiles, were all in this zone down here. We weren't seeing anything in this in vitro panel. And all of the 27 toxic drugs, which were mostly causing liver toxicity, but could also cause other types of toxicities, were all in one of the other hazard zones. And these are all widely licensed drugs that don't give clear toxicity signals in animals. So this is a panel that's actually very good sensitivity and specificity for picking up problems in humans that can not be picked up in animals. And after that, we went to a further confirmatory evaluation of 3 drugs, which we were particularly interested in because they were drugs that were endothelin receptor antagonists, and I was working for AstraZeneca, and AstraZeneca wanted to license and develop a new endothelin receptor antagonist. And we knew that there had been one drug that had been withdrawn because it caused fatal liver failure, another drug that is still widely used but with a very strong cautionary labelling for liver injury, and also a very safe drug with no evidence of liver injury. And we had a new clinical indication for this pharmacological target. And we wanted to know, was our drug going to be a safe drug or an unsafe drug? I can't give you the answer for the AstraZeneca drug because, unfortunately, for commercial reasons, the senior management decided to discontinue the AstraZeneca drug. But we did some enabling work with these other drugs to find out, could we distinguish between the unsafe drugs and the safe drugs using our panel approach? And actually, the approach worked really well. This is the withdrawn drug, the drug that never got to market, that killed people with acute liver failure, and it's the worst compound we ever profiled in our in vitro assay panel. It's a horrible disaster of a compound. This is the very safe drug, which really didn't do anything in our assay panel. Very safe drug, very good safety profile. And this is the drug that's on the market with a cautionary label for liver toxicity, which has some signals, but far less signals than the drug with acute liver failure. So that's a bit of technical data, which really brings me on to what lessons did I learn from all this complex work, particularly on adverse reactions and liver toxicity. Firstly, I was surprised that we made the arrogant assumption we could use in vitro methods and start to get some insights into hazards that could lead to rare liver toxicity in humans. And actually, it worked out very well. And we concluded that as long as we selected mechanistically relevant assays, we could generate data. But it was complicated. We needed multiple assays measuring multiple mechanisms, and we needed a way to integrate data. I've not gone into the technical details of how we analysed the data, but one of the key points was that we needed to take account of drug exposure in vivo. Now, this isn't drug exposure in vivo in animals. This is drug exposure in vivo in humans. So that's unsurprising. Potency of effects in vitro, you can only interpret if you relate them to concentrations of the drug that people are exposed to in humans. And that was a key to the sensitivity and specificity of our assay panel approach. We used mechanistically relevant assays, and we took account of in vivo drug exposure. And we had a cunning but simplistic way to integrate data for multiple endpoints. And what we were doing essentially was hazard categorization of drugs. What we weren't doing was sophisticated understanding of mechanisms. We couldn't predict whether or not an individual drug would cause toxicity in an individual patient, but we could discriminate between drugs that would cause toxicity in one or other susceptible individuals in the human population, which was all we needed to do for drug development purposes. So this actually was rolled out within the company just before I left as a way to enable us to categorize our compounds and work out, before we took them into clinical development, would they have the ability to cause these potentially concerning adverse effects later in clinical development. And senior management loved it because it gave them a way to control their investments and try and have confidence that if you take a drug into development, it's likely to be safe, versus discontinuation before clinical development because it's likely to be unsafe. So I've given, no time at all, I've given a whistle-stop tour of some of this. I just wanted to finish off very briefly by pointing out that this is just the starter, and a lot of other people are picking up this concept and running with it at the moment. There's some wonderfully sophisticated exposure-based modelling, mathematical modelling, which has been undertaken by a US consortium, the DILISIM consortium, and this is bringing in some exposure-based simulations, human systems biology thinking. And it builds on exactly the concepts that I've been describing, but a much more sophisticated exposure-based approach going forward. And this consortium has been tremendously successful in being able to predict the frequency and severity of liver injury observed in a clinical trial with a drug that caused no evidence of liver injury in animal-based studies. Triclizone was withdrawn because of liver failure, and it caused a frequency of liver signals in clinical trials that couldn't be predicted from animal-based studies, but could be predicted using this sophisticated modelling-based approach. Okay, I've run out of time. There are other approaches that are more sophisticated, building up mechanistic understanding, particularly there's the adverse outcome based approach, which is very powerful moving forward, and that's where the field is about to move to. It's difficult, but we're starting to get there. And we're also coupling up the in vitro toxicity work to in vivo biomarkers that we can measure in blood and in other ways. So there are blood-based biomarkers that are accepted for regulatory use as improvements on kidney toxicity evaluation. And I know there's a big initiative about to get underway for biomarkers that are relevant to other types of toxicities, including liver and neurotoxicity. There are also imaging-based approaches that can be used, and there's a big European initiative using whole-body imaging-based approaches, non-invasive imaging, MRI and other tracers and imaging modalities to aim to look at liver toxicity by looking at liver transporter assessment and also other toxicologically relevant endpoints. So the key thing is working collaboratively. I'm part of Safer Medicines Trust, which is one really tiny charity based in the UK. So one of the ways that we collaborate with others is via the evidence-based toxicology collaboration. And Thomas Hartung, who heads up the collaboration, will be talking later this afternoon, bringing people together so we can work effectively. And within the UK, we are forming an alliance, bringing together actual users of technology, people who hate the idea of working with animals, and scientific community and people like myself, in order to try and find better ways to develop solutions to this problem moving forward, so we don't continue to describe the problem. So those are my conclusions. I will stop now. You can read those, and if we have any time, I'll be delighted to do my best to respond to any questions in spite of my lack of German.